The overall goal of this dissection procedure is to demonstrate how to isolate and prepare the mouse intestine for subsequent characterization techniques.
This method could be used to investigate the ability of the intestinal stem cells to repopulate the intestine following an experimental procedure. The main advantage of this technique is that it can be used following any technique or any procedure or after gene manipulation using cre-lox technology.
Begin by placing a euthanized adult mouse in the supine position and wetting the fur with 70% ethanol. Then, using scissors, open the intraperitoneal cavity longitudinally along the midline and secure the stomach with forceps. Next, sever the connection to the esophagus and gently pull on the stomach to remove the small intestine up to the appendix. Then gently pull on the appendix to remove the large intestine up to the anus. Once the intestines have been isolated, remove the stomach and the appendix and use a syringe equipped with a blunt-ended pipette tip to flush the intestines with PBS. Immediately after flushing, cut the intestine into 3 equal-sized proximal, middle, and distal sections. Then cut each section into 1 cm pieces and place 3 to 5 fragments of the tissue onto the middle of a 2 by 2 cm piece of surgical tape in a pyramid formation. When all of the tissues have been attached, seal the tape around the pieces longitudinally, creating a log pile. Then place the tape in a flat-bottomed receptacle containing at least 10 times the volume of neutral buffered formalin fixative to the volume of tissue, and store the samples at 4 degrees Celsius prior to embedding and sectioning. After fixation, transfer the tissue to a flat-bottomed receptacle containing at least 10 times the volume of 70% ethanol as the volume of tissue. To fix the tissue in methacarn, immediately after flushing, cut the intestine into 3 equal-sized sections as just demonstrated. Then place each section side by side on a 15 by 15 cm piece of filter paper and use spring bow scissors to open the sections en face. When all of the sections have been split, transfer the filter paper into a glass dish containing freshly prepared methacarn for 3 to 24 hours at room temperature. At the end of the fixation, use forceps to pick up the end of each of the pieces of intestine one at a time, and wind the tissues around the forceps to form individual Swiss rolls. Secure each roll by slightly opening the forceps and inserting a 25-gauge needle through the tissue. Then place the rolled tissues into a flat-bottomed receptacle containing at least 10 times the volume of neutral buffered formalin fixative as the tissues for at least 1 hour before processing. For whole mount laxi visualization of the tissue, first pour molten raw wax mixed with mineral oil into 15 cm petri dishes. Then, immediately after flushing the intestines with ice-cold PBS, flush the tissues with 25 milliliters of ice-cold Excal fixative. After fixation, cut the intestines into 3 to 5 equal sections and place each section onto one of the cooled wax plates. Pin down each end so that the sections are slightly stretched with the mesenteric lines at the tops of the plates. Trim any excess mesentery. Then use spring bow scissors to cut the guts longitudinally, pinning as they are opened. When all of the sections have been pinned, flood the plates with enough Excal fixative to cover all of the tissues. After at least an hour at 4 degrees Celsius, use a 25 milliliter pipette or a syringe to remove the fixative and wash the tissues once with 30 milliliters of PBS. Then cover the sections with 30 milliliters of DTT demulsifying solution for a 30 to 60 minute incubation at room temperature with rocking. At the end of the incubation, remove the demulsifying solution with a 25 ml pipette or syringe and flood the tissues with another 30 ml of PBS. Then use a Pasteur pipette to remove the mucus with the PBS. After the wash, replace the PBS with 30 ml of X-Gal for an overnight stain at room temperature in the dark with gentle agitation on a rocking platform. The next morning, if the sections have developed a blue-green stain, replace the X-Gal with 30 ml of PBS. After 3 minutes of gentle agitation, make Swiss rolls as just demonstrated, and place the tissues in a wide-mouthed flat-bottomed receptacle containing at least 10 times the volume of appropriate fixative as the volume of tissue at 4 degrees Celsius for at least 24 hours prior to embedding and sectioning. Using the ROSA 26R LAC-Z conditional reporter, 100% recombination can be observed 3 days post-induction in the small intestine. Using DNA extracted from the recombined crypts, qPCR for the recombined alleles demonstrated a non-significant increase in recombination within the Lgr5 system, potentially due to the recombination in Paneth cells not observed using the cre system. Using the LAC-Z reporter, both systems also demonstrated a complete loss of recombined cells at 3 days post-induction. The mitosis and crypt cellular height data indicated the cre system could recover, presumably due to repopulation by unrecombined intestinal stem cells. In stark contrast, the Lgr5 failed to recover, despite retaining epithelial crypt cells. Further, the crypt cells in Lgr5 cat B floxed mice were non-proliferative and lacked expression of the intestinal stem cell marker OLFM4, unlike the crypts in cre mice. Similarly, Paneth cells underwent apoptosis after cat B deletion only within the Lgr5 system.
Once mastered, this technique can be completed in approximately 10 minutes. It is important to minimize delays to prevent tissue degradation. After watching this video, you should have a good understanding of how to remove the intestine from a mouse for downstream analysis.